David is a PI in the Department of Health Data Science at the University of Liverpool. He has a strong expertise in the area of approximate inference, in particular for longitudinal data modeling and survival analysis. So always with the intention to tailor these approaches to real-world settings and making them practical and efficient. And so, yes, he's going to tell us more about his recent research today. So over to you, David, if you're ready to share your screen.
Can you see that okay?
Yeah, perfect.
Excellent. Well, thank you, Helen, for that overly generous introduction. You're very kind. It's nice to be able to speak to you today, and thank you for the invitation and the privilege of speaking to you. I've followed the work of many of you in various different areas over, well, a number of years now. So it's nice to be able to speak to you and see whether some of the things today might be related to some of the things that some of you at least are working on. I'm going to talk about variational Bayes and modeling longitudinal data sets. And I should just say that if you'd like to interrupt as we go along, I'm completely okay with that. If something's not very clear, I can try and answer any questions at the end, but I'm equally happy for you to try and stop me as we go along. That's perfectly fine. Excuse me. Hopefully that's moved on for you. So I'm interested in collecting, well, modeling multivariate longitudinal data. And you'll probably be aware that this is collected routinely in lots of settings, things like electronic health records, and even quite a lot of trials collect lots of measurements of biomarkers and collect them over time during the course of a study. Now, we might want to model how a variable changes over time, or we might be interested in how some additional covariates influence the trajectories for biomarkers over time. And what is commonly done in many settings is to analyze each marker separately. So you might fit a linear mixed model for a particular longitudinal variable and analyze it on its own, independent of other longitudinal variables that you've collected. Gone too far. Here we go. Some efforts have been made to model longitudinal variables simultaneously within the same model. Most of the methods are okay at handling 2 or 3 or 4, maybe 5 longitudinal biomarkers. But even so, most methods are slow. Usually the papers will say we can handle, you know, up to R longitudinal biomarkers, but most examples, including some of my work in the past, say that, but then, you know, fit models to 3 or 4 longitudinal biomarkers, because we run into computational problems if we do anything much more substantial than that once we have big data sets. Once things get very big, we'll run into even more speed problems. Now, this is not moving on my slide, but is it on yours? Yeah, okay, so we've moved now. Okay, so increasingly we have more access to high dimensional data. I'm particularly interested in this middle point here about repeated measurements on multiple biomarkers from longitudinal studies. And I guess the end use of what we'd like to use it for are things like dynamic risk prediction. But the problems that we have are limited by computational burden. So if we just analyze a subset of variables, we might waste important information. It's gone too far again. There we go. So as a sort of introduction, what we'd typically do with longitudinal data is we fit a linear mixed model. And if we have multiple biomarkers, we might fit a multivariate linear mixed model, or if we have binary variables or count variables, we might fit a multivariate generalized linear mixed model. And the graph on the slide at the moment is just showing that the basic idea of a linear mixed model is that there's an overall sort of group profile. That's the solid red line here, the average sort of change over time in your population. But individuals might start in different places. So the dashed red line on this slide, or they might change slightly differently over time, the orange dashed line on this slide. We account for correlation between repeated measures using random effects. So random effects take account of the fact that repeated measurements on the same individual are likely to be correlated, as are repeated measurements of multiple variables on the same individual. So a common approach is to use these multivariate generalized linear mixed models. And here's the maths of the graph on the previous slide. This is just a standard setup for a mixed model. We have some predictor variables, covariates, which we're calling X here, and some coefficients beta, which describe the association between your predictors and your response, which we're calling Y, measured on patient I, marker R, and time point J. In this notation. And then we have some random effects, this U variable, and a sort of design matrix for that containing all the random effects information, Z. Okay, and we've already said we're going to induce the correlation with these by specifying a joint distribution for the random effects distribution. I've already said that this gets computationally complex as data sets get larger and we want to look at more longitudinal variables. So we might take a Bayesian MCMC approach to estimate the model parameters. If we took a frequentist view, once we get high dimensional random effects, we end up needing to evaluate quite high dimensional integrals, which is problematic. So quite often this Bayesian MCMC approach is taken. So if we set up a model according to a Bayesian specification, we're going to assume that our biomarker YR is some member of the exponential family, or we might stack all of our longitudinal variables into this common vector Y, and except that we've got some abusive notation in this line. So this B function and C function within the exponential family bracket applies row-wise and might be different for different rows depending on what type of outcome we've got, whether it's continuous or binary or a count or that kind of thing. Okay, so there's a bit of notation abuse in this slide for the sake of having something that's reasonably neat to write down. We're going to assume that our random effects jointly follow a multivariate normal distribution, and just as a sort of pointer, if we've got say a random intercept and a random slope for each biomarker, obviously the more longitudinal biomarkers you've got, the bigger this random effects covariance matrix will become. And we're going to assume fairly standard priors for mixed models in Bayesian frameworks. We're going to have an inverse Wishart distribution for the random effects covariance matrix, and we're going to introduce these auxiliary priors, these A terms, which are really only included so that we end up with uninformative priors. So that there's a Gelman paper that says that your covariance parameters should have is it half T distributions and things like that, and these auxiliary priors allow that. Then for any continuous biomarkers we've got an inverse gamma distribution for the error variance, and again some auxiliary priors just so that we can have uninformative priors. and we'll assume that the regression coefficients beta have a normal prior. So this is all a fairly standard setup of a mixed model in a Bayesian framework, nothing particularly novel here. So to estimate this model, we need an estimate of the posterior distribution of all the parameters listed here, given our data y. And my suggestion to you is that this can be complicated to do. We can try and do this using Markov Chain Monte Carlo, and that's perfectly possible, but it'll take a while in big data sets. And I'm going to suggest in this talk that variational Bayes offers a potentially quick solution, which in most cases is reasonably comparable to the MCMC answer you would get if you're prepared to wait a long time. And we'll compare at the end what I mean by reasonably good and reasonably close and potentially quick. Okay. So I'm aware that not all of you will be familiar with variational Bayes. So just as a sort of trying to demonstrate what the main idea is, I've borrowed this picture from one of Tamara Broderick's presentations at the ICML a few years ago. And the idea of variational Bayes is that suppose we have some posterior distribution we want to estimate, this p theta given y. What we're going to try and do is instead estimate we're going to approximate it using some nice distribution q star of theta. And we're going to pick our q of theta distribution so that it's as close as possible to the posterior we'd like to estimate, but we're struggling to because it's not a nice distribution. Okay, so essentially we're going to try and approximate our posterior with some product of nice distributions. I think we've maybe missed. There we go. Yeah. So mean field variational Bayes, the aim is to approximate P of all of these parameters given Y by a product of simpler distributions. So this Q of theta, we're going to split up into a product of some subsets of our parameters. And that's called the mean field product restriction. Okay, so let's consider the posterior density function P of theta given Y. So we can write that out using Bayes formula, the joint distribution P of Y and theta divided by the marginal distribution P of Y. If we rearrange that and take logs and essentially multiply by 1 on either side, but we'll write 1 as an integral of some arbitrary density on the right-hand side at least, then we end up with this equation here. This second term, integral of q theta log of q theta over p theta given y, is actually the Kullback-Leibler divergence. So that's a measure of how far our approximating distribution q of theta is from our posterior distribution that we'd like to estimate, but we're saying is too hard to estimate. Okay, so we're going to write the first integral in the previous slide, this p underlined of y and q. And then we can write that the log of the marginal density P of Y is equal to this log of P underlined plus the Kullback-Leibler divergence between our approximating distribution and what we'd like to estimate. So if we choose Q as close as possible to P of theta given Y, the posterior, then we make this P underlined term as close as possible as we can to the marginal density P of Y. Or if we think about that differently, if we maximize this P underlined term, that's equivalent to minimizing the divergence between Q theta and P of theta Y. So if we maximize this P underlined term, then we end up with a Q theta that's as close as we can get to the posterior that we'd like to approximate. Okay, it turns out that others have done a lot of work for us in this field, and you can work out the optimal form for each of your Q distributions using this equation here. It looks nice, that the maths is a bit messy, but you can work it all down, write it all down and work it all out. If you'd like an introduction to variational Bayes, you could look at this Ormerod and Wand 2010 paper, which I think is a really nice introduction to variational Bayes. And so once we work out the optimal form for each of our Q distributions, so remember each Qi is a subset of the parameters that you're interested in, a distribution for that subset, you can iteratively update them with respect to all your other Qs until there's minimal change in the log lower bound. Okay, so in my case, I need an approximation for Q of beta, that's the regression coefficients, U, that's the random effects, sigma R, that's the random effects covariance matrix, and these A terms, which are auxiliary parameters, and the variance terms for the errors for any continuous biomarkers we've got. Okay, so one way we could split up this Q into subsets of parameters is we could split the fixed effects and the random effects into one compartment, and all the sort of variance terms into another. That's one possible split. And it turns out that just because of already assumed independencies in linear mixed models, this separates further, okay, into these 5 Q terms here. So we've got a Q for the beta and the U parameters, a Q for the random effects covariance matrix, Qs for the error terms for any continuous biomarkers you've got, and Qs for these auxiliary A parameters, okay? So all we need to do is work out the optimal form for each of the Q densities. It turns out that's particularly messy maths, but a lot of variational Bayes more or less boils down to adding 0 or multiplying by 1 in fancy ways. So you can work out the optimal form using the equation we had on the last slide. If you have conjugate priors, everything usually works out quite nicely. So it turns out that 4 of the 5 Q terms that we have in our model all have nice, easy updates. Once you do the maths, you can recognize it as some known distribution that's easy to recognize and has known updates in our algorithm. If we only have continuous longitudinal biomarkers, then even the Q beta U term as well has a nice easy update and everything works nicely and easily. But if we have binary longitudinal markers or Poisson responses, we end up with a problem because we can't recognize the optimal form as some nice easy distribution, a normal distribution or a gamma distribution or something of that sort. These log terms or exponential terms cause us problems when we work through the algebra. So what we've done to overcome that is we've followed an approach called semi-parametric variational Bayes, which was proposed by Rody and Wand back in 2016. And we essentially specify that this particular Q distribution has to be a normal distribution, and we pick the parameters of the normal distribution to be as good as possible, or optimal in some sense. An additional problem we have when we have binary longitudinal biomarkers is that we end up needing to evaluate this integral here. There are a number of approaches you could take to do that. You could do some quadrature, or you could do some approximation somehow. There's an approach by Jacola and Jordan to do this. The approach we've taken in this paper is to use Monahan and Stefanski approximations. So this is just a very fast numerical approximation to this integral. We've taken the view that since the point of variational Bayes is to get an answer fast, we may as well make a little bit more of an approximation along the way for the sake of speed. And so we use these Monahan and Stefanski approximations to evaluate this integral here. And Nolan and Wand prove some results to show that this is not an entirely terrible thing to do, and give some bounds on the approximation. It's pretty all right. Again, oh, I missed a slide. Yeah, the updates for the Q beta U term still end up with a problem because you need to find the inverse of a very large matrix, especially when the number of patients is large. But it turns out that the matrix that we need to invert is sort of block diagonal. It has the structure shown on the slide here, and there's lots of sort of zero chunks to it. So in fact, we can take advantage of a matrix algebra result from this textbook by Harville that gives you how to calculate the integral for that. And it means that you only end up needing to take inverses of much smaller matrices, this sigma q beta term, which is essentially the dimensions of this are related to the number of fixed effects you have. So it's far smaller. And so you can get your updates much more quickly, and the computation is faster. So we end up with an algorithm for our variational Bayes way of approximating multivariate generalized linear mixed models. The question then is, how do we tell whether it's any good? I haven't presented the algorithm in full here. It takes about a page of the paper that it's published in, which I'll mention to you at the end. So you'll just have to take my word for it at the moment that I have an algorithm that I'm claiming is a variational Bayes way of estimating multivariate generalized linear mixed models. How do we tell whether it's any good? Well, we can compare the posterior distributions we'd get using variational Bayes with the posterior distributions we'd get using Markov Chain Monte Carlo for models and sample sizes where that's a feasible kind of thing to do. And so I'm going to calculate what I'm calling the accuracy, which is essentially the difference in the areas under the posterior distributions, the non-overlap, if you like, in the posterior distributions. Okay, so I'm going to calculate the density of an MCMC output using a kernel density estimate with sort of standard plug-in bandwidths, and I'm going to see how close my posterior distributions are to those I would have got if I'd done MCMC. Okay, so first some simulated results. I considered 3 different sample sizes, 100 individuals, 1,000 individuals, 10,000 individuals. And in the first set of simulations we had 3 Gaussian longitudinal responses with the equations here and the random effects covariance matrix given by U here. And then in the second set of simulations, I have 1 Gaussian, this Y1 here, Y2 becomes we'll specify as a binary longitudinal variable, and Y3 as a count. So we'll just put a logic function in front of E of Y2 and a log in front of E of Y3. Okay. And so these simulations show that the fixed effects parameters, the accuracy terms were all pretty high for the fixed effects for these 3 Gaussian markers. Okay, so the variational Bayes estimates of the fixed effects were pretty close to the Markov Chain Monte Carlo estimates of the fixed effects. Things are less good for the covariance matrix parameters. So there's worse accuracy over to the right of this top plot, and you get the same kind of thing in the second plot where we have 1 Gaussian, 1 Poisson, and 1 binary longitudinal variable. This is a known kind of problem with variational Bayes, that we don't estimate variance parameters very well quite a lot of the time. The sort of the mean value or the mode of your posterior distribution is usually pretty close, but we usually don't estimate the variance pretty well, and that seems to particularly happen for these covariance parameters. This problem gets better if you have more repeated measurements per individual. So the accuracy is better the more repeated measurements you have per individual. That I think makes sense and is probably not very surprising, but is at least reassuring. But can still be a problem if you have 3 different types of markers. So once we have binary and count markers, the accuracy is less good for a lot of the parameters that are associated with those variables in particular. But still the fixed effects are reasonably well accurately estimated. Excuse me. So next I'll move on to some real data applications. I'm going to apply my algorithm to two data sets. The first is a commonly used longitudinal data set that's available in R, and it's to do with patients who have primary biliary cirrhosis. This is available in quite a lot of R packages. It's in the MixAK package, it's in the Joiner package, well, Joiner ML, I think, and JM Bayes and packages like that have this PBC data in it. So there were 312 patients, and there were 7 continuous longitudinal markers and 3 binary markers. The details of them are not especially important for this example, but we'll say there are 7 different continuous markers and 3 binary markers. We're going to adjust each of our models for age and sex and the type of drug they received. So people were either on a placebo or D-penicillamine in this data set. And we're going to include fixed and random intercepts and fixed and random slopes in each of the longitudinal submodels. So the point of this PBC data is to try and assess the performance of variational Bayes compared to MCMC in a setting where MCMC is perfectly feasible. Even the most complex models on 312 patients aren't too much of a problem to estimate with MCMC. That's perfectly doable. So we're going to compare our model results where it's perfectly feasible. Then the second comes from a screening trial that we were involved in here in Liverpool, individualized screening for diabetic retinopathy. So individuals who have diabetes turn up once a year for a screening visit where their eyes are, they take an image and they grade, and they're trying to identify people who are high risk of sight-threatening diabetic retinopathy, which can cause blindness. So as well as their screening markers, which we're classing here as 2 binary markers, one for each of the left and right eye, there are 10 continuous longitudinal markers, things like HbA1c and blood pressure, HDL cholesterol, LDL cholesterol, and a number of other longitudinal biomarkers. The trial was trying to find good ways of screening high-risk patients more frequently and low-risk patients less frequently. For the purposes of this, I'm just using the data to fit a multivariate generalized linear mixed model in quite a big data set. So there were 16,133 patients in the data I'm going to show you in a minute. And we're going to fit mixed models for each of these outcomes, which correct for type of diabetes, age, duration of diabetes, and the sex of the individual, and again have random intercepts and random slopes in our model. Okay, so the first thing we're going to do is compare, going too far. Yeah, the first thing we're going to do is compare the speed of our model fitting. So in the top part of this table, we've got the time in seconds for the PBC data. And you can see that even the 10 marker model, essentially I'm just increasing the number of markers each time. Even the 10 marker model with MCMC, 3,000 seconds, it's about 15 minutes or so. But it takes about 15 seconds using the variational Bayes algorithm, so it's quite substantially faster, but still MCMC is feasible in this situation. For the diabetic retinopathy data, in the bottom part of the table, we've now got time in hours, and to fit this full 12 marker model, so in this case, you've got a 24 by 24 random effects covariance matrix, 62 hours, not quite 3 days, but getting on towards that, and variational Bayes takes less than an hour. So again, quite a substantial speed up using variational Bayes. Something that would take a few days can be done in less than an hour. Now, with all of these things, speed comparisons are subjective. It depends on how many samples you take for your MCMC. For these, we did 5,000 burn-in and kept 10,000 samples, I think, was the settings. And it also depends on the stopping criteria for the variational Bayes algorithm. We stop when the change in the log lower bound was, I think in this case, less than 10 to the minus 7. So depending on what stopping criteria you use and what number of samples you choose for MCMC, you would change the figures in this table a bit. You might be able to write an MCMC algorithm that worked quicker than the one that's built into the MixAK package in R, if you're good at MCMC algorithms. So you could maybe improve on the MCMC time. Stan was slower, but you might be able to improve. And well, equally, you might be able to write more efficient variational Bayes code than me. But nevertheless, with all of these caveats, variational Bayes seems to be quite substantially faster than using MCMC. And you can see that this might be an advantage, especially when you get more than 16,000 patients and lots of longitudinal markers. Of course, speed comparisons are only helpful if what you're estimating is accurate. So here's a few example slides. This first slide is from the PBC data. And we've just picked the 3 biomarker model just for the sake of having something that would fit on a page, but you could look at output for any of the models and get something pretty similar. So we've got fixed effects. We had an intercept and a drug and a sex and an age and a time fixed effect. That's each of the columns here, and 3 biomarkers, each of the rows here. In blue is the MCMC estimate, and in orange is the variational Bayes estimate of the posterior distributions, and I've put the accuracy score on as well. So you can see here that more or less all of the fixed effects, the posteriors that you get with variational Bayes, are pretty much the same as the posteriors you'd get with MCMC. Same kind of thing on the next slide. We'll go back one. For the ISDR data, again, I've just picked the 3 biomarker model. So there's 3 continuous biomarkers in this. But again, the MCMC and the variational Bayes posteriors are more or less identical. If you looked at the covariance matrix posteriors, you would see less good accuracy, but still you'd see the sort of modes of these distributions in roughly the same place. They wouldn't overlap as well, but the fixed effects, which is often what people are interested in, are certainly well estimated. As I said, it's quite hard to display all the results from a 12 marker model, but here's a small attempt at it. So here we've got fixed effects for intercept and time for each of these longitudinal variables, HbA1c, cholesterol, and so on, all accurately estimated except the 2 binary ones. Something has not worked well here. And the covariance matrix in this B plot, the green is more accurate. Okay, so the more green these plots are, the more well accurately estimated the variational Bayes posteriors are compared to the MCMC. And in plot C, what I've done is I've converted the covariance matrix, the estimated covariance matrix, into a correlation matrix, just to try and show that one of the reasons we might model everything simultaneously is to try and see how the different biomarkers are related and correlated. So the sort of more strongly red or more strongly blue squares in plot C are things that we might pick up on and see stronger relationships between different longitudinal variables. Okay, so the take home, I think, is that variational Bayes estimates fixed effects parameters pretty accurately, estimates covariance matrix parameters less accurately, but still not terribly in this case, and we can fit it in less than an hour instead of 2.5 days. In this plot, we take 3 particular patients, shown by the orange and the green and the blue points and lines. I'm not sure how well this will come across on your screens, but the dashed line is the variational Bayes predicted profile for that particular biomarker and that particular patient, and the solid line is the MCMC predicted profile for that patient and biomarker, and these basically overlap. We haven't put confidence regions on to try and keep the plots tidy, but they would overlap reasonably well as well. Some of the work I'm doing at the moment, which I haven't got time to talk about today, when I'm not doing COVID research and stuff like that, my variational Bayes interests at the moment are whether we can improve this estimation of covariance parameters. That's a known fact of variational Bayes, that it sometimes doesn't estimate covariance well. Sometimes that doesn't matter much, but when it does, there might be ways that we could correct our estimates. I've listed 3 here: Pareto smoothed importance sampling, linear response variational Bayes. These are things that people have suggested in some settings of correcting variational Bayes estimates so that you get good covariance estimation as well. And that's something that's sort of ongoing work that hopefully might improve my variational Bayes algorithm a little bit. Directions of extensions that we could take with this, particularly in respect of things that I know some of you in Cambridge are very interested in. I'm currently thinking about extensions to this work in terms of adding a survival component so that we could have multiple longitudinal biomarkers and time to event data. Again, there are some packages in R that do this with MCMC approaches, but they're going to have computational problems if you get very big data sets and lots of longitudinal markers. I think variational Bayes offers an opportunity there. Adding in the survival component has currently proved trickier than I have managed to solve so far, but that's ongoing work. And also some of you are working on location scale models and things like that. I think variational Bayes would work well in that situation to additionally model the variance terms in the model. I think that would be an interesting extension. I've listed some other things that I think are worth thinking about, but I haven't done any work myself on. One of the limiting factors of variational Bayes is that it doesn't give you the theoretical guarantees that a lot of other approaches might do. There is some work improving theoretical understanding of variational Bayes. I think more could be done in that area, and it would be useful if it was. Variational Bayes is used in loads of applications. It would be good if there was some results underpinning that, or more results underpinning that. And it might be that in my work, we've used the Kullback-Leibler divergence, which is standard for variational Bayes, to choose our Q distributions. It might be that you could do better if you used different divergence metrics. I know some people work on stuff like that. I think it probably makes the algebra a bit more tricky, but there may be possibilities of improving variational Bayes or getting better theoretical results if we were to go down those lines as well. So a summary of the work I've presented probably far too quickly. Variational Bayes offers a faster way of fitting multivariate generalized linear mixed models. And in general, the accuracy of the estimates is pretty good. Variance estimation is better if we have more measurements per individual. And I suppose the advantage of modeling everything in one simultaneous kind of joined model is that we can consider the correlations between lots of markers all at the same time. That could be interesting. If you're interested in looking at the algorithm in more detail and the sort of maths behind it all, you could look at our paper, which was recently accepted in biostatistics. The reference is there on the screen. And obviously, I'd be happy to answer any questions you have. And as always, this was not work solely done by me. Co-work with Marta Garcia-Fignana here in Liverpool and Matt Wand in Sydney at the University of Technology. So I'm grateful to them for their help with this work. So thank you for your attention, and I'd be very happy to take any questions you might have.
Thank you, David. Very, very informative and interesting talk. Thanks. So are there any questions for David? I think there is one in the chat by Filippo. So I don't know, Filippo, if you want to unmute yourself and ask your question, or otherwise I can read it.
Yeah, I mean, the question has been answered during the talk. I mean, you said you used Stan, right? So I'm assuming you used HMC.
Mm-hmm. Yeah. So I used Stan, and I've also used the MixAK package in R, which has multivariate generalized mixed models, which I think is broadly similar to Stan. I've not got all the exact details, but essentially HMC.
Yeah, and I mean, while I'm here, following up on that, I saw that you showed the discrepancy on the marginals of the parameters that you estimated. I was going to, this is a comment or a suggestion rather than a question. I was going to suggest that it's going to be a lot of plots to look at, but it's also very useful to look at the triangle plots where you look at the 2D marginals of the whole distribution, because in case there are some funny correlations going on, that's where you can see if things are going wrong. And if not much is going wrong, then you can use that to lend additional support to the method that you're using.
Yeah, that's a good suggestion. Thank you. That's not something I've especially done, but yeah, that's it.
Yeah, no problem.
Okay, there is another question, Jessica.
Hi, David. And thank you for the nice talk. So I remember seeing you talk about this kind of in an earlier stage at the Channel Network conference a few years ago. So I just wondered if you could say a bit more about the extension to joint modeling with a survival outcome. So what are the kind of additional complexities there?
So at the moment, the additional complexity is that when you do the formula that gives you the optimal form of an update doesn't have a nice recognized form. So I think essentially, again, you have to end up making this assumption that you're going to pick the best normal distribution that gets close. And most of my attempts so far have run into numerical problems when I try and optimize it. So the exponential term in the update essentially gets too big too quickly, and I've not yet found a really solid way around that. So there are a couple of things we've tried. You can put like a threshold. So if the exponential of the bit that you're taking the exponential of gets above some safe threshold, you can just cap it, and that sort of works okay. Other than that, I'm a bit stuck for ideas at the moment. So essentially, you can work out the maths, but then when you try and run the algorithm, I run into numerical issues.
I see. Yeah, because I think it would be really valuable to have.
Yeah.
And as you say, the joint modeling packages that exist at the moment are really quite slow when you try and scale them up to data sets and more markers and so on.
Yeah. Yeah, I agree. So if any of you are experts at optimization and numerical tricks, I'd be very glad to talk to you and see if any of you can help me.
Yeah, I'd be interested in the extension to location scale models as well, if you ever get onto that.
Yep.
Thank you.
Sean, you had a question?
Hello, David. Yes, thanks very much for the talk. Sounds very useful, this algorithm. I just wanted to ask two questions. Firstly, I presume that you can handle situations where not all of the markers are observed at the same times. And secondly, you say the method works best for the estimating the fixed effects, posteriors of the fixed effects parameters. And I just wondered whether, I mean, how much efficiency do you get in estimating those things, those fixed effects, from modeling the outcomes jointly?
Yeah, that's a good question. So the first question about whether everything's measured at the same time is not a problem. In the diabetes example I showed you, not everything was measured at every visit. Some of them were measured at most visits, and some of them definitely weren't. So that is not a big problem. The question about what, if we're only interested in fixed effects, what do we gain by modeling it in a multivariate model? I don't have an answer for that in terms of a table that I could show you. There are a number of papers that sort of suggest that, you know, you can get better predictions if you consider multiple longitudinal biomarkers. You can sort of handle correlations better by modeling everything together. Verbicki and a few others have a review of multivariate mixed models, which I think delves into this question a bit more than I've been able to answer here. I guess it depends what you're looking for. Univariate models might be perfectly fine depending on what the question is you're trying to ask. So you might not need a multivariate mixed model, but that's certainly true.
Okay, thank you.
Maybe if I can follow up on Sean's question. So I've seen it in the estimated correlation matrix you showed. There were a couple of markers, pairs of markers that showed very high correlation, like LDL and cholesterol, I guess, which is expected, I guess.
Yep.
And so, but also most of the pairs seem to have quite low correlation, right? So I don't know if you tried, for instance, based on the continuous markers that you model, try to predict the retinopathy scores, so the binary scores that you also have, and try to see whether modeling jointly these markers improves the prediction performance when compared to univariate modeling or univariate based prediction of this score. I don't know.
Yeah, that's a really interesting suggestion, because Peter Diggle has a paper where he does a multivariate model for diabetes. I think it's for diabetes. He doesn't have it's not a mixed model he fits, but it's some kind of multivariate model that he essentially uses as a missing data prediction kind of tool, which is a bit like what you're suggesting, I think. And the advantage of modeling it all together is that you can then predict missing values or unknown ones. I haven't done any work on that, but that is quite an interesting suggestion. And essentially in joint models, that's sort of what's trying to be done. You're using the longitudinal predicted values maybe to go into your survival part component and predict time to event outcomes.
I see. Yeah. Okay, thanks. Sheila, you had a question?
Yes, thank you very much, David, for a lovely talk. Harking back to COVID, I think the diagnostic agreement between the methods that you have, even for the variants, people would be delighted with for LFTs versus PCRs. in the COVID setting, so that, I mean, the sort of 50% agreement, agreement isn't so low for something as difficult as variance. But I wondered whether there was any potential to be gained from using more patterning in the priors about which variances you might expect to be the same, so that you can borrow strength from different parts of the modelling.
I think my internet dropped out for the first part of that, but I think you were essentially asking whether you could choose your priors more carefully or cleverly to get better variance estimation. I think that is a good suggestion. Essentially, everything we've done in this piece of work is to use uninformative priors, as that seems to be sort of the standard thing people do in these Bayesian mixed models. If you had reliable ways of choosing better priors, that probably would overcome some of this. One of the last suggestions on my slide of ways to improve covariance estimation was to remove some of the auxiliary parameters. So that's the bit that makes you have this non-informative prior. So some of Matt Wan's early variational Bayes stuff didn't put those variables in, and it might be that if you sort of take them out again, you might do better by having, well, cleverer priors, essentially. I'll look into that.
Okay, I see there is a question in the chat by Javier. What kind of survival models are you considering for the joint modeling extension?
So at the moment, only fairly simple ones. I'd like to look at Cox models, but at the moment, most of my work were things like Weibull models, you sort of fairly standard time to event kind of survival models. Most of the variational Bayes work that has done any kind of survival modeling, of which there's not very much, does kind of neural network approaches to survival analysis, which I have to say I don't completely understand. So I've not seen any variational Bayes that does sort of standard parametric survival modeling before. They all do neural nets, but the stuff I've tried and failed so far with has been Weibull models, gamma models, fairly standard things.
Can I ask a question, David?
Yep.
Have you considered subsampling your data set to have a stochastic variational inference?
No, so I haven't. So what is the main idea of?
Like you're evaluating the likelihood on the full data set, and you're trying to scale on bigger data set, but like it's trivial to extend mean field variational Bayes to use just a part subsample of the full data set, and that kind of gives even exponential speed up. So like I think good references like Matt Hoffman's work on that. So it's called stochastic variational inference. I was just wondering, because you're trying to scale to even larger data sets. So have you considered that?
Yes. So I haven't so far. One of the things I'm sort of interested in is whether we actually need huge data sets. If you could just take a reasonable subset, maybe a clever set and a clever subset in some way, you might not need to be able to fit complicated models to, huge data sets. So maybe that's a way to go. And people do that with MCMC in a number of different ways, don't they?
I think a lot of the work on Bayesian core sets are kind of in that direction. So like I think Tamara Broderick has done some good work on that. So. Yeah. And I have one more comment actually about your figure you represented, where you showed like the variational distribution within the big posterior distribution. I have just one question, because at the end of the day, to have the KL or to compute the KL, you need to share the support of the distributions, right? So if the distributions don't share the support, then I think the KL is undefined. So that figure, it was a bit confusing to me.
Okay. So my simplifying figure ended up not being so simplifying after all. I'll try and think of a better one for the future. Thank you.
But that's just a comment, yeah.
Yeah.
Are there any more questions? Yes, Tweet.
David, thank you for a great presentation. Really interesting work. The computational comparisons were very impressive. I was wondering if you had to use something like C++, or did you just use standard R programming to get those very fast variational inference timings?
So the variational Bayes was entirely stuff just in R. So there was nothing fancy than that. I think the MixAK package for the MCMC stuff has some C++ calls that it makes, and I honestly don't know what Stan does in the background in their algorithms. But the variational Bayes stuff was all just stuff written in our standard sort of matrix calculations.
And just an extra question, if I can. Mm-hmm. Have you considered an extra level of hierarchy? So what you've got so far is a longitudinal model where you apply it to each specific patient or subject, but we could do things like, group subjects into age brackets or something like that, and that would create an extra level of hierarchy. I was wondering if you've considered that, and if that would fit nicely within the structure that you've used already using these fast covariance matrix estimations.
Yes, so I haven't. I know you have a number of nice papers working on that kind of thing. So that would be something I'm interested in doing, because you could feasibly sort of consider patients within hospitals with electronic health records and things like that. I haven't come across an example where I've needed to do that so far, but that's an extension I'd certainly like to work on. That'd be interesting. Just need a good example to justify it.
Thanks again.
Thanks, Pete.
Any more questions? Doesn't seem to be the case. I think anyway, we are reaching the hour now. So yes, I'd like to thank you again, David, for the very interesting talk that raised a lot of questions. So it was very interesting. And yes, thank you everybody for attending, and have a nice afternoon.
Thank you.
Thank you.